Hi everyone. We just finished chapter 8 talking about reproduction and how cells divide. And so we're kind of going to extend that and move from just like how to create like cells from to like how do parents share their characteristics and their genes with their children. And so we're going to be studying inheritance. So the study of genetics has a lot of historical roots. Genetics here refers to the study of how traits are inherited from one generation to another. And so in the past, one of the first people to think about this concept was someone named Hippocrates. Hippocrates, and he had a lot of ideas. A lot of them weren't correct, but he was one of the first people to think about this. He kind of thought that genes, the parents' genes or traits would blend together for the children. And so what that meant was, like, for example, if there was a short mom and a really tall dad, then their genes would actually blend together to create like a new gene for the child, and all the children would be like medium height. And so he thought this happened for everything. Like the genes would alter and like kind of mold together, almost like clay that you squish together with another piece of clay and you create like a new color. We actually have found through modern science that that is not true, but that's kind of the thoughts that they used to have. But the fundamental question that we're going to be exploring is like, how do characteristics get passed from the parents to the children? So if it's not this blending model of genes blending together, then how does it happen? So the history of genetics, as we mentioned, heredity is just the passing on of traits from one generation to another. And genetics is the study of that process. It's the study of heredity. The first person to really kind of establish this field and to like figure out some of the key concepts of it his name was Mendel, Gregor Mendel. And he's considered like the father of genetics just because he did so much work in it. And he made his discoveries while studying the plants in his garden. So Mendel, this guy, he argued that there were certain heritable factors, which what we call genes. He didn't know that they were genes at the time, but these heritable factors retain their original identities. So in contrast to the old blending model that they used to have, what he believed was like the genes of the parents, the father and the mother, would kind of come together for the children, but they wouldn't change their identity. They would still stay the same genes. They would just get put together. And so the genes would retain their original identities. They would still be the same genes. They would just be combined together. So that was kind of like the new idea that he introduced. So what are these heritable factors or features? So there's something called character, which is kind of like a characteristic. This is review of what we talked about before, but a character would be like any heritable feature that varies among individuals. So examples of that's like flower color would be a character, flower shape, like any kind of characteristic that you could use to describe something is probably a character or heritable factor. And so within the character, you can have different manifestations of that characteristic. You could have different traits. So traits are just the variation of that character. So if the character you choose is flower color, then the traits of that character would be like the flower color could be white or it could be purple. If the character was flower shape, then the traits could be like it could be round or it could be pointy. So this diagram just shows you all the different kinds of characters you could have. You could have height, seed shape, seed color. All of these things are different characters. And then over here, the different traits. You could either be tall or you could be short. So these are the different traits that you could have for each character. So Mendel, how he kind of discovered these different principles of genetics, what he did was he was breeding plants until he made sure that he had true breeding varieties. That means that his plants, the way he started off with, they were purebred, kind of like pure dogs, where you know that they are like their parents are the same breed, and then their grandparents are the same breed, so they're pure. He did that with his plants, so he can kind of start off with the same basis. So he had true breeding varieties of the parents, and then when he combined them together, he would create hybrids. Hybrids are when you take two different varieties and you combine them together. True breeds are when like the parents are the same and their grandparents are the same. And so he would do these many generations of like breeding. So P generation is like the parents, the true breeding parents. And then F1 generation is like the first generation. So that's like the children of the parent, P generation. And then F2 generation, that's just the offspring of the F1 generation. So you can think of P generation kind of like the grandparents, the F1 generation would be like the parents, and then the F2 generation is like the children. Or you could think, yeah. So he discovered a lot of different things using something called a monohybrid cross. A monohybrid cross is when he would breed like two plants, but only like examine one character. So for example, you can see here that this is a picture depicting color. So the character that he was studying was the color of the plant. And over here represents its genes. It had like one gene that was like represents big B and one gene that was like little b. And he crossed it with another plant that was big B, little b. And then he kind of figured out what the offspring was like. So we'll talk a little bit more about monohybrid crosses, but this is kind of just an introduction of what they look like. He took two like plants and he would breed them together and like look at the offspring, and that's how he discovered all of these different things. And so here is a diagram from the textbook. What he did was he took purebreds. So like this purple flower, it was like a pure purple flower. Its parents were purple, its grandparents were purple, its great-grandparents were purple, and then this one was pure white flower. And so he took these two flowers and then he bred them. He put the seeds together to form the F1 generation. Remember, the F1 generation is just the offspring of the P generation. And so he noticed that all of the children or the offspring of these two, the purple and the white flowers, would be purple. And so he took these purple flowers from the F1 generation, and then when he bred a purple flower with this purple flower from the F1 generation, he found that he got purple and white again. So he wanted to figure out why this was the case. And remember, he was just looking at the physical color of the flower. He didn't know what the genes actually had because he couldn't analyze those. And so based on Mendel's experiments, these are the different principles that he was able to discover. The first thing he decided was that for every one character, so that means like flower shape, flower color, for every one characteristic, there could be alternative versions of the genes for that character. And so these different alternative genes, which really code for traits, are called alleles. So alleles is just the word we use to describe these alternative genes you can have for every one character. Something else he said, so for each character, an organism has two alleles. So you get one allele from the mother and one allele from the father. And that really makes up remember those homologous chromosomes we've talked about before, how each homologous chromosome would be coding for the same characteristics, but they would have different traits. The new word we're introducing is alleles. Alleles is those genes that code for those different traits. Now, if the alleles from the mom and the dad are the same, so like let's say, for example, the allele for eye color, let's say it's blue and blue. If they're both the same, then the organism is considered homozygous for the gene. That means that the two alleles are the same. If the alleles are different, like blue eyes and brown eyes, then that means that the organism is heterozygous. So these are two different words to describe homozygous. The two alleles are the same. Heterozygous means that they're different. If the two alleles for a character are different, then what is the one that you actually see? What's the one that determines the appearance? The one that determines what the organism actually looks like is the dominant allele. So let's say you have two different alleles, one for blue eyes, one for brown eyes. The brown eye allele tends to be dominant, which means that it will overpower the other allele, and all you will see is brown eyes. The allele that you can't really see if there's if you have two different alleles, that one's called the recessive allele. So the blue eyes would be recessive, the dominant allele would be brown because you only see brown. Mendel also discovered something called the law of segregation. This is really important. it just basically says, it's not super complicated. The law of segregation just says that each gamete, each sex cell, so either the sperm cell or the egg cell, only carries one allele. Um, so remember, you start the parent cell was like that had the full number of chromosomes, but that chromosome number got divided in half to form the haploid gametes, right? So the homologous chromosomes got separated, meaning that each daughter cell, each sperm cell or egg cell or whatever gamete only has one allele. But then the alleles, the one allele from the sperm cell and the one allele from the egg cell will combine together again during fertilization to form to form the offspring, and then the offspring, which develops into a full human being, all now have the full two alleles for every one character. Okay, so Mendel did something called, we talked about the monohybrid cross. This is just a way that Mendel used to analyze kind of what he observed. And so I'm just going to describe how to make a monohybrid cross. You start with something called a Punnett square. Punnett square is just like a square with 4 boxes inside. And so what you do is you put the parent alleles on the top and on the axes, on the sides. So let's say you start off with a parent that's big B and big B, so it's a purebred. Let's say big B stands for brown. So it has 2 brown eyes alleles, so that would go on the top. And then on this side, let's say these are the mother genes, the mother alleles, and so the mother is blue-eyed, and so she has 2 little b's. So you start off with putting the parent alleles on the sides, then you carry the parent alleles down and you put 1 in each square. So from the father, big brown eyes, big B, you put 1 big B in each little box. You're just writing the same B down in each column. And then same thing with the mother, you write the little b straight across. So here you just completed your Punnett square. Every one of these little squares represents the genotype of each offspring. Genotype is our fancy word for the genetic makeup, the genes of the possible genes of the offspring. And so in this case, all of these happen to have the same genotype, which is big B, little b. That's the genotype. And now the genotype affects the phenotype of the organism, and the phenotype is just the physical traits. And so in this case, we said, let's say big B, oh sorry, this should have been brown, but big B is brown eyes, which is dominant, then the phenotype would be brown, even though there's a little b in there for the blue eyes, you can't see it, so the phenotype would be brown. Sorry, that should be brown, not black. Okay, the last section here is just reviewing homologous chromosomes. This is kind of review. But remember, every diploid cell, so every cell in your somatic cells or in your body, have a pair of homologous chromosomes. So 2 chromosomes, they kind of code for the same characteristics or characters. One of them comes from the mother and the other one comes from the father. So in your body right now, you have a set of genes from your mom and a set of genes from your dad. And each of them, you have pairs of them, these chromosomes, and they code for the same characters. And notice at the same locuses or loci, remember that's the same location on these genes, these homologous chromosomes, it'll code for an allele. So at the specific locus, you have the 2 chromosomes have alleles for the same character. Now the alleles can be the same as in here, and if they're the same, remember it means that they're homozygous, and it could also be different. So the allele from your mom and the allele from your dad could be different, meaning making it heterozygous. So there's a lot of terms in this chapter. Just kind of make sure you review all the terms, all the bolded words, and then we'll talk more about this in class. Thanks for watching.